Good morning, everyone. It's a real pleasure and an honor for me to be here and kick off the scientific part of this wonderful meeting. And I'd really like to start by thanking the families for their resilience and motivation, and by sharing that your commitment to improve the life of your loved ones is really one of the biggest source of inspiration for the work that my lab does every day. So really, thank you so much for that. So today I'm going to tell you about early function of Syngap1 in the development of the human brain and enteric nervous system. And my laboratory has been focused on really improving modeling of human brain development and disease. And particularly, we've been focused on understanding intellectual disability and autism spectrum disorders. So despite the fact that these disorders are, there has been a huge effort from the field in trying to understand these diseases, we know that in the last 70 years, we haven't really been able to come up with effective treatment for these disorders. Obviously, there are many reasons for this, but to me, something that is actually very important is that these disorders have a very complex underlying genetic. So most of these disorders are actually polygenic, which means that the same variant in the same genes, but in two different individuals, can actually lead to a completely different clinical manifestation. So this has obviously complicated a lot our understanding of these disorders and our ability to come up with effective treatments. So this really, to me, speaks for the fact that we need to complement the work that the community has been doing with the animal model system with human tissue. So we really need to study human tissue to dive into the genetic of these disorders. So ideally, what we would like to do to understand this disease is to sort of go back in time and look at how the brain of a specific patient developed in the womb. Obviously, we cannot do that, but human brain organoids that are derived from pluripotent stem cells really, as they have brought us closer to this ideal scenario. And they really have opened a window into aspects of human brain development and disease that we haven't been able to model before. So brain organoids look like these. So you can see in this image. So here there is a PhD candidate in my lab that is holding this flask in which we grow organoids. So they look like lentils. They are about 2 millimeters wide, big. And they are derived from pluripotent stem cells that can be derived from somatic cells of specific patients, from either blood or skin. And then we get these cells, we revert them to pluripotency, and then we are able to differentiate them into brain organoids. So there are different protocols to differentiate brain organoids. One that is unguided, that generates organoids that contain different brain regions. In our lab, we use guided protocols that allow generation of organoids from specific brain region. And so in particular, we are using these telencephalic organoids to model and understand a bit more about Syngap1 function in human brain development. And these telencephalic organoids generate cortical tissue. And we know that the human cerebral cortex is the structure in the brain that is responsible for the execution of higher order function of the brain that is really, these are the functions that make us human. And we know that these disorders affect specifically those functions. So we generate cortical organoids, and this is, if this works, is you can see this is a reconstruction of how an organoid looks like. So you can see here in red these progenitors that line these ventricle-like structures that really resembles the ventricle in our brain. And then on the top of this ventricle, you can see these neurons that are lying on top of the germinal zones, much like they do in the actual human brain. And so, yeah, we know a lot, obviously. We have had a wealth of knowledge that has come out from studies in the last 20 years or so about the function of Syngap1 in excitatory neurons, mostly from mouse models. And my lab has recently discovered expression and function of Syngap1 in in human cortical progenitors. So obviously also in the human brain, Syngap1 is expressed in neurons, but we found this additional function and expression. This work was led by Marcella Birtele, an amazing postdoc in my lab, and in collaboration with Marcello Coba. And so as you can see, this is a section of a cortical organoids. So you can see here in green these germinal zones where progenitors are localized, and these small ventricles that are these fluid-filled cavities that resemble the ventricle in our brains. And so you can see the Syngap is expressed in neurons, but also in this, around this ventricle. So you can see here Syngap expression. So this is called apical end feet of this progenitor. So Syngap is here. And so these cortical progenitors that are called radial glia are very important cells and have this very special type of morphology. So you can see that they are bipolar. So there is one process that contacts the ventricle and one that is the base up to the meninges. And so basically, when there is decrease in the level of Syngap1, we saw disruption of these interactions. And radial glia cells are very important for the generation of the cortical column in the cortex. So the cortical column and the basic unit of our cortex. And so if the contacts with the ventricle or the extension of this process are altered, what happens is that there may be problem in how these cells divide and how neurons walk and migrate on this basal process. And so really in the way our cortex is built. And so we have already, and this is published, so we have shown that in cortical organoids, when there are decrease in level of Syngap1, the distinction that we usually see between progenitor zones and neuronal layers, that is also obviously there in human tissue, and we see in control organoids, is disrupted. And you can see that now neurons are actually present in this progenitor zone. And also when we look at division mode of these radial glial cells, how they divide, we see a disruption, and these radial glial cells start making more differentiative divisions, and so they start making more neurons instead of making more progenitors. And along with this, we found that neurons when levels of Syngap1 are decreased, differentiate faster, they are more complex, and they have higher level of activity. So this is all published already. And while we know, we were able to publish this new novel function of Syngap1 in cortical progenitor, we really don't know much about the mechanism and the signaling driven by Syngap1 in these progenitors. And so I'm happy to actually announce that we have just received confirmation from the NIH that we got a big grant to pursue the studies of this mechanism in progenitor cells. And this is collaboration with the Coba lab. And so we'll be able for the next 5 years to do substantial work and understand a bit more about this signaling. And what we are also doing, thanks to seed funding from the Syngap Research Fund, we are trying to understand whether other variants are also leading to the same type of phenotype, the developmental phenotype in Syngap1 organoids. And so the first study I show you was focused on this variant in the RAS gap domain of the protein. We have repeated some of this work in another cell line with a variant in the DAF domain of the protein. And for this, we have done work on an assay that is called a Rosette assay. So in this assay, which is very high throughput and faster than a normal cortical organoid assay, you can see that there is generation of one lumen, and then radial glia are organized around this lumen. And so if there is a problem in how these radial glia develop, how they extend their processes, you can see it very easily. And so in our first study, we show that this variant in the RAS gap domain leads to this disruption in how this rosette can form. And now we repeated this also for this other variant, and we saw something that is very similar. So it appears that actually also variants in the DAF domain, this variant has the same type of phenotype. And we have also done the same work in organoids and look at the functionality. And we have seen that indeed there is an increase in functionality also for this other line. And so these are, we are trying to do more work on these. And we would like to extend this work to other variants. So we have been, we have a big grant pending with CERM in which we are trying to characterize 30. So we are proposing to characterize 30 different variants and characterize the developmental trajectory and electrophysiological properties of organoids derived from these 30 patients, Syngap patients with different variants. And so SRF has been amazing in supporting us throughout this application. So hopefully we'll get this funding because obviously this type of work requires extended funding. So we hope we will be able to do this. And now I'd like to switch gears in the last couple of minutes and talk about a new direction that is being spearheaded by Marcella Birtelen in the lab. And this is a project that Marcella is really close to her heart. And so please, if you want to know more about this, go and talk and interact with Marcella, that is actually sitting here in the audience. And so Marcella became interested in understanding the role of Syngap1 in the development of the enteric nervous system, actually after joining this same conference last year. So talking with families, she realized that actually how pervasive gut issues are in this patient population and how disruptive these issues are. And then we were delighted to see that a few months ago, Ellen Wilsey's lab published a very beautiful work in bioRxiv about this topic. And she also did some work and analyzed some data and showed that looking at citizen dataset and Simon's collection, that indeed for many of these genes associated with autism spectrum disorder, there is many of these patients have these very pervasive gut issues. And it's also evident that when we look at the expression of autism genes, and in particular Syngap1 in gut, in the gut, we can see that they actually are expressed in enteric neurons, and in particular subtype of enteric neurons. And so what Marcella decided to do was to generate these human enteric neurons from pluripotent stem cells using this protocol published by the Studer Lab in 2019. And she, sure enough, found that a very similar phenotype to what we have found in cortical neurons is also present in enteric neurons. So we see accelerated development of this neuron and increase in the level of activity. And she has also now worked on other of modifying this protocol, and she has seen that in spheroid that derive from pluripotent stem cells and that contain these enteric neurons, she sees problem in the migration of neurons that have a delayed migration time, and then also increase again in the activity of these enteric neurons that carry these variants in the RAS-GAP domain. And so now basically our plan is to also try to see if these alterations are present in other variants. And also Marcella has started co-culturing this enteric neuron with intestinal neurons in collaboration with Jason Spence at the University of Michigan. And so we want to see how these alterations in Syngap enteric neurons may also disrupt the intestinal niche and the property of these intestinal organoids. And so thank you. So again, I'd like to thank the people in my lab that actually did the work, and Syngap Research Fund for funding our research and supporting us in all possible way. And thank you for listening.